want to start by giving everyone a really warm welcome here tonight. My name is Maike. I'm a physician and I'm a scientist, and I'm the director of the Pediatric Diabetes Research Center here at UC San Diego. And looking at this almost full room here, I want to actually express my gratitude for so many of you taking the time tonight and actually coming out to join us for this event. Our audience tonight is actually pretty diverse. I know that many of you are personally affected by diabetes, either yourself or your family members, but then we also have people in the audience who came here to really hear about the scientists from local biotech companies, and then also from the various academic institutions here on the San Diego Mesa. So one thing that unifies clearly all of us today here is that we want to really see a game change in the therapy of diabetes, that we really want to see after years and years of waiting that a change is finally on the horizon. And this stem cell clinical trial, which is the first really trial involving stem cells for the treatment of diabetes, could really be this game changer. So it's incredibly exciting times right now. And in the more than 20 years that I have actually worked on diabetes and been in diabetes research, I've not seen anything of this magnitude ever come really into clinical trials. So it's incredibly exciting times, and it's also exciting that it's taking place right here in San Diego. And this is not really a coincidence, because we have an incredible community here. We really have the best science here and amongst the best scientists and the best environment to really make something like this happen. So this is why I've actually brought my colleagues out here tonight to tell you about the various pieces of the trial, and to also tell you how to put something like this on really takes a collaboration between many. So not like a single institution or individual can actually achieve something like that. This trial has a history of almost 20 years of work that has finally led us to actually take cells into patients. So what are the pieces that actually takes to make this happen? There's a lot of basic research that went into really understanding how these pancreatic beta cells that we're trying to put into people now are made. And then it took a company, so an industry partner like ViaCyte, to take this knowledge that was mainly generated by people in academic institutions such as UC San Diego, and then take that and translate that into something more applied. But then industry can't really put on a clinical trial. For that, they once again actually need partners, so they need a place where there's the best doctors that really have the knowledge of the disease that then can partner and really take these things into clinical trials and make sure these trials are conducted ethically and safe manner, because we all want that these trials are safe and we don't want to really rush into anything. With that, I also want to kind of caution you tonight that it's really early days for this trial. I know you're all eager to ask a lot of questions, but it is still very early days, and for a lot of your questions there might not be answers yet, but feel free to voice it all. We want to actually hear from you and hear all the things that you have on your mind, and our speakers will try to answer them the best they can do and the best they're able to with the knowledge we have so far. So one thing that it then takes, and the industry partners and trials partners, it also takes then people in the community, actual advocates who are champions of the disease, and we have people here in San Diego who have been true champions. For example, Lorraine Steele, who's been tireless really advocating for diabetes. Then we have philanthropists who've really helped us to really actualize sort of our ideas and to help with the research and help with the clinical translation. And then in the area of diabetes, Lisa and Steve Altman have been incredibly generous and dedicated here. And then also David Winkler, who was actually the one who helped found the Pediatric Diabetes Research Center. And this is just to name a few, and I know we have many more in the community. So I'm really excited to also announce that actually the Dean of Health Sciences, Dr. David Brenner, has recently announced that he will make diabetes a true priority for this university. and that he wants to build a comprehensive diabetes center here at UC San Diego. So what we hope to actually then have in the center is that we can actually have all these puzzle pieces where you have the science and then you have the industry partner coming in, the clinical translation, sort of have a frame for all of this where we can put this together, where the scientists can work alongside the physicians who actually understand the disease, and then these teams can then help translate new knowledge that arises at the laboratory bench right to the bench site. And we also want the center to be a comprehensive clinical care center, so that if anyone in the community is diagnosed with diabetes, they can actually, as a child, then find the best physicians who help treat the disease, and then that should be a seamless handover then to also the adult clinical care, so that there's really a comprehensive care for diabetes. So without further ado, I then want to turn actually the podium over to the first speaker, Dr. Larry Goldstein, who is the director of the Sanford Clinical Stem Cell Center here at UC San Diego.
So I'm Larry Goldstein. I'm director of the UC San Diego Stem Cell Programs, and more recently I'm director of the Sanford Stem Cell Clinical Center, which has worked with the company ViaCyte, the other diabetes units at UC San Diego, the Pediatric Diabetes Research Center run by my friend and colleague, Dr. Maike, to put together what I think is really a groundbreaking effort in science and medicine. There are two reasons to love this clinical trial. One is that it's going to make a real difference for our understanding of disease potentially. The other is that, as near as I can tell, it's 100% homegrown in San Diego. So first of all, I'm an alumnus of UC San Diego. Paul has been a member of the faculty at UC San Diego. Kevin DeMoore, one of the lead scientists, was a graduate student. I think I was on his thesis committee when he was a student here. And many of the folks who work at ViaCyte who have made this into reality come from the local San Diego community. And so it really exemplifies the way we do things here in San Diego, a group of people, a group of institutions working together to make something really extraordinary happen. And I'll come back to that in just a moment. Now, the other thing I'm going to do that's a little unusual is it's very unusual for scientists to talk about clothing. We're generally characterized in the media and the cartoon pages of the newspaper as having no sartorial discretion whatsoever and not even having a clue as to what to wear. And that's actually true for myself as well, but my wife makes sure that I dress properly before I leave. But there is one piece of clothing I'm going to talk about this evening, and that is my tie. Now, the reason I'm going to talk about my tie is that it has little diagrams of the polio virus on it. So you may remember that 50 years ago, the polio virus was the scourge of humanity, crippling and killing children almost without reason and direction. It was horrifying, terrifying for people who dealt with it. And yet, that challenge was possible to beat. It was beaten by a combination of groups working together in spite of the competitions you've all read about. And it was defeated with great science. And of course, that's the key, ultimately, to really defeating a terrible disease such as type 1 diabetes. You got to bring great science to the party, and great science working with people from industry and from clinical medicine will win out. That's what it takes to defeat a disease. And that's what we're seeing happen here, even as we speak, with the ViaCyte stem cell clinical trial. Now, the history, for you history buffs, is we've known of the pretty amazing technological capabilities of stem cells for some years. The debate about human embryonic stem cells started almost 20 years ago in 1998. And through a steady application of basic and applied science, these embryonic stem cells from humans have been gotten under control in a way that my colleague, Dr. Paul Laikind, will tell you about it in a few minutes when he gives you the nuts and bolts of how this particular trial works. But then there's another facet that's happened here in San Diego, and I'm going to use a cliche, unfortunately, and that is it takes a village. It's often used, usually abused, not used. In this case, I'm going to use it because it has taken a village to develop this trial. So the UC San Diego stem cell program started in 2006. I was its first and still far only surviving director. The Sanford Consortium for Regenerative Medicine, the building you're in right now, was set up to bridge from basic science into translation from bench to bedside, and then most recently the Sanford Stem Cell Clinical Center, set up with a truly generous gift of my friend Denny Sanford, has set up the clinical infrastructure from which we can do one thing and one thing alone, and that is to accelerate stem cell clinical trials. Whatever it takes to get good science with stem cells into clinical trials, such as the clinical trial that we're discussing tonight, developed by the company ViaCyte. Now remember, it's a safety trial, it's early in the game, but the fact is that this village of entities, the various stem cell programs, ViaCyte, our partners on the Mesa, the Pediatric Diabetes Research Center, and then ultimately the Altman CTRI, these have all been key pieces for bringing together what is really, when you get right down to the nuts and bolts, very tough science and very tough clinical medicine. My friends know that I often say, problems in human medicine are hard. They really are. All the easy ones have been solved with antibiotics. And what we're at now is a time when we really have to dig in and work hard to solve the more difficult problems of type 1 diabetes and Alzheimer's disease and Parkinson's disease and many, many others. But tonight is a watershed in that it's really our first public discussion with all the players present about what it's taken to get this first clinical trial with stem cells for type 1 diabetes up and running. And so I think that's really all I want to tell you this evening. I can take questions from you at this point, but I'll just reiterate, great science makes great medicine, and it takes a village of us working together to make this happen. Maike.
So now we would actually like to hear from you. So is there any questions that have already come up that you might want to ask Dr. Goldstein about regarding the Sanford Center, the stem cell vision that we have here at UC San Diego? If not, I know you're probably all eager to actually hear about the clinical trial.
Well, they're here for the beer.
So with this, I will turn the microphone over to Dr. Paul Laikind, who is the Chief Executive Officer at ViaCyte, which is the company that actually laid the foundation for this trial here in San Diego.
Okay, well, thank you. So it's a real pleasure to be here to talk to you about something that is really exciting, really important to myself, but also to a very dedicated team of people, certainly at ViaCyte, but at UCSD, at JDRF, at the California Institute of Regenerative Medicine, and within the whole diabetes community. We're so excited about the work we're doing there, and so it gives me pleasure to tell you about it. I got to say, trying to do it in 5 minutes is a real challenge for any CEO, but I'll do the best I can. So, as I said, I'm really here representing a very dedicated team at ViaCyte of men and women that have been working for over a decade to take some of the work that, as was said, from understanding what the disease type 1 diabetes is and turn that into a treatment. And the treatment we're working on that I'll tell you more about, if it is as effective as what we've seen in the animal models, and that's always a bit of an if, but one that we're pretty excited about, if it is as effective, then it would essentially be a cure for type 1 diabetes. And I'll tell you why that we think that going forward. So, first off, I think, as was said, a lot of you in this room already are touched by type 1 diabetes, but I'm not going to make the assumption that everybody is. And so let me tell you a little bit about the disease of type 1 diabetes, because it also sets up how we are looking to treat this and give this potential functional cure for it. So type 1 diabetes is an autoimmune disease where your body, or the body of patients with type 1 diabetes, recognizes the beta cells not as self, but as an invading cell, and the immune system essentially destroys those cells that are responsible for producing the molecules, the hormones that regulate blood glucose. And if you can't regulate your blood glucose, this can lead to many complications, even death. And so it's very important to be able to control that. So in type 1 diabetes, this autoimmune disease occurs. The beta cells, which is the cell that produce insulin in the pancreas, are destroyed by the immune system, and this leads to the disease. When you're unable to produce that insulin, you can't control it. Before there was insulin, if we think back around 50 years or so to before insulin was brought out as a pharmaceutical product, type 1 diabetes was a death sentence. Okay. Today, insulin allows us to control the disease, but it's not perfect. It's far from perfect. There's still long-term complications. There's still a lot of issues with hypoglycemia associated with insulin. So we're continuing to look for better ways to manage this disease. What we're doing at our company is developing a compound or a product, or a product candidate at this point, I should be careful to say, called VCO1. Not a very exciting name, but it's what we use. VCO1 is a stem cell-derived islet replacement therapy. So as I was just mentioning, in the disease with type 1 diabetes, the beta cells, which are part of the islets, a collection of cells in the pancreas, are destroyed. And so we're looking to replace that with VCO1. The way we do that, the way we replace these cells, as Larry was already talking about, is we start with a stem cell starting point. In our case, we start with an embryonic stem cell. And it is a embryonic stem cell line that we harvested a number of years ago, and we have banked that cell. And so we start from a single source of embryonic stem cell. We then go through a process called differentiation, where we take that embryonic stem cell and we scale it up. We go from millions of cells that we take out of the freezer. We can scale that in a couple weeks to literally billions of cells. We then go through a process of differentiating those cells from that embryonic stem cells up to a pancreatic progenitor cell. And that sounds easy. It takes about 12 days to do that differentiation, but I can tell you it took about 8 or 9 years of work to do it. And again, as was alluded to earlier, there's a lot of looking back at what goes on in the basic research world to understand how cells differentiate in the body, and that we use that to then design the techniques and mechanisms to do this outside in a manufacturing setting. And doing that, going from that embryonic stem cell up to that pancreatic progenitor cell is not rocket science, but what is rocket science is doing it in a regulatory compliant way where you get the same product out each and every time you do a manufacturing run. And that's where 7, 8 years of work and about 50 patents all concentrate on. So that gives us a cell product where we can go to these pancreatic progenitor cells, but we need to put that into a patient. These are human cells going into a human, but they're not the patient's cells, and therefore you can have an immune reaction just like you hear in transplant surgery and such. You can have an immune reaction to that transplant. So we need to protect those cells. And so a second part of our technology is called the Encaptra drug delivery device. And this is, think of it as a tea bag, where we put these cells in this tea bag with a semi-permeable membrane. It allows the free flow of nutrients and proteins, but it doesn't allow those cells out of the device or any of the host cells, any of the immune cells into the device. And this has been shown in multiple models to be protective against both allo and also the autoimmunity that's common in type 1 diabetes. So we take these cells in this tea bag-like device, and this is put under the skin in various locations we could use. And then the idea is they will be vascularized, the cells will begin to grow and mature, and they will become, over a couple-month period, they will continue to differentiate and become, for all intents and purposes, islets. And actually, Maike did some of the seminal work to help us understand that and show that that is indeed the case. So once they're in the animal, or hopefully in the human, and they have differentiated into these cells, they then can sense the glucose in the blood and release insulin, glucagon, other regulatory hormones in a regulated fashion to control that disease. And so what we're hoping as we treat these patients, that as those cells mature, they require less and less insulin to control the disease until by the time they have fully matured, they can throw the needles out the window and no longer need to use insulin to control the disease and be essentially, in a sense, functionally cured for type 1 diabetes. So we're very excited by what we're doing in this area, but as it was said, we certainly have not worked in isolation. We have worked with many scientists throughout that decade of work. We built it on the basic research from institutions like UCSD, the Pediatric Diabetes Research Center here, and others around the world, similar institutions around the world. And we've worked with leaders like Maike and Larry and many others to understand and develop this product to the point where we can move it to the clinic. You know, as Larry said, it takes the best, to get the best medicine, you have to have the best science, and that is absolutely true. You cannot skimp. You've got to make sure that this is not only an effective, but also, of course, a safe treatment before we go into the patients. I do want to say that another component of allowing us to get to where we are is that we've had tremendous support from the patient community, but also both financial and technical support from advocacy groups for diabetes and for stem cell research, including JDRF, as well as the California Institute for Regenerative Medicine. We could not have done what we've done to date without those organizations. They've been critical to allow us to get to where we are. Obviously, we're a private company, so we're a for-profit company, so we've also had investors, and I don't want to leave them out of that equation. They also are important for getting where we are. But, you know, now that we have gotten to this stage, when we did a lot of work, and last year we started putting together our application needs to go into the Food and Drug Administration. We started putting that together and getting the preclinical work in place with anticipating going to the clinic. We got that IND approved, and we essentially walked across the street to UCSD to begin the clinical trial. And while geography is great, the reason we did that wasn't just geography. It's because we really had the best team available right here in our neighborhood to carry out this trial. And so we were very excited to do that. We have, you know, doing a clinical trial, you become much more outwardly focused. As was mentioned, we can't do those clinical trials ourselves. We need to work with outside groups. And UCSD has been fantastic as we have moved into the trial. Right now, we started this trial towards the end of last year. We currently are in a subtherapeutic dosing of it, so we're looking at safety. We've done 4 patients to date, and we're continuing to enroll additional patients in this initial safety part. But once we have demonstrated that the cells are working the way we hope they are and that it is safe and well tolerated, we hope to move into a second part of this trial where we will go to a fully efficacious dose and look not just for safety, but also efficacy. And the endpoints in this trial are very clear because we're looking at patients that don't produce insulin, and once we treat them, if they now do produce insulin, it's pretty clear why that happened. So working with people at UCSD, our PI, our principal investigator is Bob Henry, a past president of the American Diabetes Association. We work with Jeremy, we work with James Chow, just a really top team here at UCSD. You know, just in closing, I just want to say, you know, there's no guarantees in clinical research. You know, this is a tough nut to crack, translating from animals to humans in type 1 diabetes is a very challenging area. But one thing I'm very confident of is that we've got the right people, the right team, the right clinical setting, and I believe and hope it's proven that we have the right product to really make a difference for this disease. And I'd be happy to take some questions.
Thanks a lot, Paul. So I'm sure there's already the first question.
Thank you very much for your comments. I just want to ask
Please
you.
use the microphone.
Oh.
Yeah, that everyone
Okay.
can hear.
I just want to know, how has your experience been, or ViaCyte's experience with working with the FDA? Have they been more obstructive? Have they opened the door?
I've been in the biotech industry for almost 30 years now and worked with the FDA many times. I have never been in a more cooperative situation with the regulatory authorities as we have at ViaCyte. Now, I hope I can continue to say that over the long term as we continue to make progress. But we worked with the FDA. We did a pre-IND meeting. IND is the investigational new drug application that you have to file to get permission to go forward with the trial. We did a pre-IND meeting with the FDA. We did a pre-IND meeting with them. At that pre-IND meeting, they basically offered up something that I've never seen them do, which is to work with us in real time to allow us to send in protocols and such. And that seems like, why wouldn't you do that? But that, the FDA has a lot on their plate and generally don't do that. But they have worked very closely with us. Our IND application was almost 10,000 pages. This is a very complicated product with both a device and a cell therapy. And we put that in and got it approved in 30 days. So, we're approved for, I should say, approval means we got the green light to go ahead with the clinical trial.
That's encouraging, for sure.
More questions?
Lisa?
Yeah, please.
Oh,
And
sorry.
Lisa, or so front, and then.
What was I have laryngitis, sorry. What was the timeline that you saw with these in the animal studies, the progenitive cells, to start producing insulin?
Yeah. So we put in pancreatic progenitor cells. These are cells similar to what's in an embryo that goes on to form a pancreas. And the reason we choose those cells rather than going all the way to a beta cell and implanting those is when we first put these cells in under the skin, which is where we place them, they're in a hypoxic environment. They have low oxygen in that environment. And these embryonic cells are designed to exist in that kind of environment and to recruit vasculature. So that's what we see in the animals. And to answer your question, we see it in about 2 to 3 months for the cells to get vascularized, to begin growing and filling the device and producing insulin. We don't know whether that's the time frame it will be in humans. That's based on the animal studies.
Is another question?
Hi, I'm a mother of 2 type 1 diabetics, and I have a question about like saving a type 1's cord blood. Is that effective, or a siblings? For those of the parents out here that are still planning their families, should they save cord blood for the future babies?
I think I'm going to put Larry on the spot and ask him to answer that one.
This one comes up quite a bit, not surprisingly, but I can tell you 2 things. Well, 3, okay. So first, most of the time that you put cord blood away for your child, it is very rare that that child will develop a disease that they can be treated with that cord blood. Second, most of the cord blood in this country is nonetheless locked up in private cord blood banks, but it's actually the public banks that do the bulk of the cord blood transplants, usually for diseases of the blood. Leukemias, thalassemia, sickle cell, diseases of that sort. There are ideas about how you might use cord blood to perhaps treat type 1 diabetes. They're very early on the drawing board. In my personal opinion, they have nowhere near the power of the approach that ViaCyte has taken with Paul Laikind's leadership.
Maybe as a quick add-on as somebody who actually studies the beta cell. So people have tried to make beta cells from many sources, and really these embryonic stem cells that ViaCyte are using that suited cells where it's been shown you can really direct them to become pancreas for cord blood cells that has not been shown yet.
Thank you.
One thing I want to take an opportunity just to make a point. You hear a lot about stem cells out there. With a lot of groups that are out there, you hear about stem cell treatments in Mexico and things like that. Those are very different than what we're doing here and what is being done at UCSD and such, where we understand the mechanism of what we're doing. We're actually not implanting stem cells. We start with an embryonic stem cell as our starting material, but when we implant it, it has been differentiated to a pancreatic progenitor cell. So it's no longer, quote unquote, a stem cell.
Some more questions here?
No, I was also, I guess, the question would reflect back to Dr. Larry. I, too, am a mother of 2 children with type 1. And with our 3rd child, we core banked her blood. So what about her blood for her older 2 siblings? I assume, hearing your response, it would be the same.
Yep.
Yes.
Thank you.
You have the microphone and a question there and there.
Thank you. My question is for you, and are there any age restrictions with this clinical trial?
Right now there is. Right now the age restriction is adults 18 to 55, I believe it is. 18 to 55. This is the first trial ever of a therapeutic, a stem cell-derived pancreatic progenitor cell replacement for diabetes. It's the first time this has ever been done, so we're going slow, and we're taking all precautions to ensure the safety of the patients, which is paramount. So right now, it's restricted to that age group.
Thank you.
So, in an ideal world, sorry, over here, if everything goes well during the clinical trials, when is your expected product to market? And especially for kids.
Good question. It's very hard to predict. One of the things we're looking at is we're looking at strategies that would allow us to not only target the kind of what I'll call typical type 1 diabetics, but also target higher risk patient populations, like what's called hypoglycemic unaware patients. And there we believe we can potentially petition the FDA for breakthrough product designation, which could allow for an accelerated development path. But even with that, I'd say you should consider it in the neighborhood of 3 to 6 years, depending on how everything goes.
Taking one last question, and then maybe for the next round, save your question.
I have a couple specific questions, which I'm not sure you're able to answer. But how long has it been since the first person was implanted with Encaptra now?
It was back in end of September, right? October 22nd. Yes.
So at this point, we'd see. At this point, you'd have an idea if the vascularization is taking as long as you thought, or if it's happening the way you thought. And
You're right.
you give
I
us?
can't talk about these. It wouldn't be appropriate anyhow. We're talking. When you're talking about the first patient, you're talking about one patient, right?
Right.
Okay, so, you can't draw any conclusion from such small numbers. We need to accumulate more and more data, which was what we're doing, and plus we don't even have all of the analysis done on what we have done with that patient. So, and keep in mind, this is a subtherapeutic dose, so we don't expect to see any evidence of efficacy, certainly at this time frame, and even out a longer period of time in this first cohort.
Do you have any idea how many devices will be needed in order to bring someone to normal glucose control?
Well, the answer to that question is if you're talking about on a drug development or a commercial basis. A commercial basis, we're expecting it, assuming everything works the way we expect it to, which is always a big assumption. But assuming everything works the way we expect it to, we're expecting to have 1 to 2 devices implanted to give a therapeutic dose. In the clinical trial, it's more than that because we're doing dose ranging, and rather than having multiple sizes of devices, which would be multiple applications and such, we have a single size therapeutic device that we can use multiple devices to do different doses. So in the trial, there's more than 2 devices implanted.
So with that, thanks for all your questions, and for all of you whose questions we haven't been able to kind of bring to the podium, please save them. There will be more opportunities, but for now, I want to turn the microphone to Dr. Jeremy Pettus, who is an endocrinologist here at UC San Diego. So he's actually somebody who takes care of people with diabetes as a physician, and he is actually the physician involved in the clinical trial, and he will tell you a little bit more about the details of the trial and where we're at.
All right, thank you. So I was really nervous sitting here and watching Larry and Paul talk, without a podium. I feel very naked up here. Like I need to do a stand-up routine of some kind with this single mic. But anyway, so I'm Dr. Jeremy Pettus. I'm an endocrinologist at UCSD. I've been living with type 1 myself for 20 years now. I just had my 20-year diaversary a couple months ago. And so, it's actually very interesting for me personally to be part of this because, I remember when I was diagnosed, having my physician at the time tell me, they're working on this encapsulation thing. They're using seaweed to encapsulate beta cells, and don't worry about it. We're going to have a cure in 5 to 10 years, this kind of thing. So to kind of go through my life as I normally would as a patient, and to be here 20 years later as a physician, working on this study is just, it's unfathomable. I just feel like a kid in a candy store that I'm so lucky to be, a part of all of this that we've all done. It's absolutely mind-boggling to me. And on cue, I can feel my continuous glucose monitor reminding me that I have diabetes because I'm nervous and my blood sugar is now alerting me that I still have diabetes. So anyways, we got work to do. All right, so I don't ever write notes, so you know that I'm taking this seriously. So anyways, who am I? So I'm the endocrinologist, one of the endocrinologists working on this study. The principal investigator, as we mentioned, is Dr. Bob Henry. I work very closely with him, and Dr. Steve Edelman, that's here as well, working on the study. And my specific role is recruiting patients, finding these people to be involved in the study, and I kind of guide them through this entire process from an endocrinology standpoint. What's their insulin capacity before putting these devices in? What is it afterwards? This is a 2-year study that they wear the devices for, so this is a very long time period that I'm involved with these people. And I know them very well, and I see them every day, or not every day, but a lot. So let me talk about what the study is, because we've been talking a lot about the background, and there's been so many years of work going into this. Let me describe exactly what it entails briefly, but let me just bear with me because I think this is very important. So as we've mentioned, there's 2 different sizes of devices. There's what I call the large device and the small device, okay? So the large device is about the size of a credit card. It's about that size and about that thin. And that is what I call kind of the workhorse. That's the one that has the majority of the cells that we're going to implant in people. And then there's the smaller device that's about the size of a dime, okay? So in this first cohort of patients, we're putting 2 of the larger devices in their back and actually 4 smaller devices, okay? And so this gets a little complicated, but bear with me. So we're putting them in patients' backs, okay? And generally on one side of their back, we put the 2 larger devices. And on the other side of the back, we put the 4 small devices. So the 2 larger devices, again, are the workhorses, and they stay in the entire 2-year period, okay? They're the ones that we're really seeing if they're kind of doing anything. And the 4 smaller devices are dime-sized devices that we call sentinel units that are designed to come out at later time points to really look at what the cells are doing. Because as we heard already, that these aren't fully differentiated beta cells and alpha cells and everything yet. So those are designed to come out as a safety monitoring to look at them under the microscope and see, are these becoming insulin-producing cells? Is there an inflammatory response? What's the histology doing? So that's kind of how it works. So on day 1, when we put these devices in, we put the 2 larger devices on one side, the 4 smaller devices on the other side. The 2 large ones stay in for 2 years, and the 4 other ones come out at pre-specified time points. At 4 weeks, 12 weeks, 26 weeks, and then sometime later, again, to see kind of what's going on as a safety monitoring process. So people say, why do we put them in people's backs? And that's kind of a fun story. So there's nothing special about the back per se, other than it's kind of out of the way. So ViaCyte has done some actually neat studies looking at how kind of robust these devices are, because the last thing you want to happen is for them to kind of rupture and for the cells to get loose and get killed and all that kind of stuff. So they actually implanted these in cadavers. They put them in cadavers, and they literally hurled baseballs at them at 60 miles an hour. I think it was 60 miles an hour. And they didn't rupture. They didn't break. So the cells, like these devices, are pretty hardy. They don't, we have people in this study that are surfers or mountain climbers, all these kinds of things. The last thing we want is for these people to change their lives because of putting these devices in. So they're very hardy. They don't get, just broken up if you get tackled in the back or you take a surfboard in the back. But they're very susceptible to a piercing issue, namely an insulin syringe or a infusion set or continuous glucose monitor. So we want to put them in the back that's out of the way of where you would put your infusion set or your CGM or any of that kind of stuff. So that's why they're going in patients' backs. Okay, let me refer to my notes. Okay, so we put them into people, and the study takes, or sorry, not the study, the surgery itself takes about 45 minutes, an hour. We actually have one of our plastic surgeons, Dr. John Walensky, who's here. John, say hi. That's actually been doing these in some of our patients to put these devices in. We've been doing them under general anesthesia, actually at the Thornton Hospital here. And so far, everyone's gone through this procedure safely, come out of it, basically on top. And it just goes right kind of under the skin. And then what we do is we just kind of follow these patients and we see, is there any signal in terms of the insulin production? And interestingly, as we mentioned, that these become fully functional islet cells. So not only are they beta cells that make insulin, but also alpha cells that make glucagon. Perhaps this could help people with issues of low blood sugar, which all of us with type 1 diabetes or treating type 1 diabetes know that's a huge issue. So anyways, the surgeries have gone well, and we've done 4 patients so far. We have our 5th patient lined up to happen in the next couple weeks. I don't want to identify them individually, but 3 of our 4 patients are here today. And I just wanted to give them a big round of applause. And, they're really the heroes of all this. And, for them to go through everything that they've gone through, and this has never been done in humans before, I'm, just immensely proud of all of them. So I think that's pretty much all. I didn't expect to get choked up over this, but I feel very responsible for these people, so thank you very much.
Feel free to ask your questions. Yes, you're not quite a point yet.
Put you on the spot again. A couple of questions. One is, I'm guessing one of the patents is the tea bag that these are put in, and I guess it would be a semi-permeable membrane that allows no autoimmunous, it doesn't allow the immune cells to get through. Have you found a problem with pocket or capsule creation around those tea bags, so to speak, that create an issue with vascularization?
So again, you could take that one, yeah.
First off, we can't say from the human studies at this point, we can't talk about what's going on there. In the animals, we do see a foreign body capsule formation around them. The fact that these are active cells inside, that's a typical thing with medical devices. You do get this foreign body response and you get a fibrotic capsule. So we do expect a fibrotic capsule to form in the humans, but what our hope is what we see in the animals is that it's a well-vascularized capsule. Okay, and we believe that comes about because of these pancreatic progenitor cells that are inside the device are releasing factors that promote blood vessel formation. So unlike a typical inert medical device, we're hoping to see that vascularization.
And the vascularization isn't actually through the membrane, I
It's
would say.
not through the membrane. If you look at micrographs from the animals, you can see that vasculature is lying right up alongside that semi-permeable membrane, but not going through it. And that's why, as he said, the devices are very thin, as he said, like a credit card thinness. It has to be that way because we have to have the diffusion distance for the oxygen and the nutrients and proteins to be able to diffuse in and out across that membrane.
Questions?
So would the ultimate goal be for just to have the 2 large devices? And how long are you thinking that might last someone? A year, 2 years, indefinitely?
This is something that, I also wanted to address, is that people always ask, what are the expectations of this study? And what I've heard is that we anticipate that, of the actual study devices that we're using, it would take 6 to maybe 8 of these devices for someone to become completely insulin independent. So putting 2 of these devices, 2 of the larger devices in a patient to start with, is a purposeful kind of underdose, because this is all about safety. You know, this is the first time it's ever been done in humans. So the first thing we want to make sure is when we're putting these into people, you know, is there going to be an autoimmune response, a foreign body reaction, all this kind of thing. So it's really just kind of about safety. Did that answer your question? Yes. Okay. And so, and for how long it'll last, that's something that we really don't know. Because a lot of these studies, again, were coming from mice. And, you know, the lifespan of a mouse in the mice that they typically did this in was about a year. You know, and those devices typically, correct me if I'm wrong, would last the whole lifespan of a mouse, you know, but then the mouse dies and you're left wondering how long is this going to last in a human. So there's a lot of things that we're still trying to figure out.
Devices are what last, the cells in the devices what we look at.
Right.
And we still see function out at the end of life of the mouse. We don't know beyond that.
Did the mice need anti-rejection drugs with a human?
They do not, but it's a kind of a trick answer, okay? Because when we do these studies in animals, in humans, when we put these cells into a human, as I said, they're a human cell, but they're not the patient cell. That's what's called an allogeneic graft, or an allogeneic, and you get an allogeneic rejection from the immune system for that. When you put a non-human cell into, or a different species cell into an animal, okay, so in our case, a human cell into a mouse, that's what's called a xenotransplant. And that's a much more robust immune response, and the device is not designed to protect against that. So we use immune knockout animals, and that's why we're limited to the mouse studies that we do. So the answer is we don't need any immune drugs in the mice, but that's because they have no adaptive immune system intact. We have done those studies, we and many others, not just us, with our device and other macroencapsulation devices have done studies using various species, baboons, using one baboon's fibroblast transplanted into a second baboon with a macroencapsulation device. Done that with monkeys and with rats and mice and various species, and shown that it will protect both allo and autoimmunity. Actually, there was also a small study with parathyroid cells in humans in Scandinavia that showed it would protect. So there's good evidence it should protect without the need for immunosuppression, but we can't say that based on the animal studies alone.
And I would add to that, importantly in this clinical trial, we're not using any immunosuppressive medications when we implant these in the patients.
So with this, do you want one last question?
Okay.
And then
That's fine. I'm a father of a 2-year-old type 1 diabetic. He was diagnosed at 16 months. And one of the big problems that we have is because of his age, whether it be FDA or insurance regulations, is getting a CGM approved or getting a pump approved. Can you just shed any light on age restrictions? I assuming the 4 patients you've had are all adults. Do you have any advice on children getting this in the future?
maybe that's actually a good segue into our last speaker, who is actually Dr. Maike Gottschalk, who is the Chief of Endocrinology at Rady Children's Hospital. So maybe Mike can actually then address some of those questions also with regard to children, and we'll maybe save that for sort of the last question answer. So with that, Mike.
Thank you. Well, I know the hour's getting late, so I'll skip the 150-slide presentation I had. But we also want to emphasize, besides the great science, also the great clinical work that's going on at UCSD. And our vision is to really develop a true comprehensive diabetes center for not only for children, but for adults. As Maike alluded to, one of the things that we are going to work on over the next year is to develop a transition clinic, because as we move children into adulthood, the type of care changes, particularly in the way the patients are cared for. So for those who've been at Rady Children's or have had children who've gone through our system, you know it's very child-focused and family-focused. But when they become young adults and they see Dr. Pettus, now they're sort of stuck because he starts asking the questions and they're like, well, why are you talking to me? Talk to my phantom father who's behind me, who normally speaks for me when I go to clinic. So we have to prepare them for really the adult world to become responsible for their care, and anywhere from, you know, how do I fill a prescription to what does my health card look like, where is my health card, how do I make an appointment. So those are things that we want to work on So we too can make a seamless transition in their care. The others really want to sort of power the great resources we have both at Rady as well as UCSD in terms of our diabetes care. There's no point duplicating resources. We really want to bring the two together to utilize the synergy that we have. In the pediatric facility, we have a really comprehensive care system because not only just physicians, but more importantly, we have nurse educators, we have dietitians, we have social workers. We were fortunate this last year to get support from a grateful family to now hire a full-time psychologist who works with us. And so we want to bring all of that activity up here to UCSD as well. So we're looking over the next year or so of moving the clinic and really having a true diabetes clinic with both children and adults can have comprehensive care. I think the other aspect that we have, which hasn't been talked about, is we also do train the future physicians and scientists here at UCSD. And so both in the adult program and in the pediatric program, we have fellowships for endocrinologists, and so we are training individuals such as Jeremy, who came through the adult endocrine program. And so it's a great pipeline for us for the future physicians and scientists who are going to continue to take on the work and legacy that those of us who are here will pass on to do that. I think going back to your question about, when are children going to be next in line, that's always the question we get asked. And as it's been already stated, safety is the primary importance. So certainly the FDA will not allow any study done with a drug or a device until there's been proven documented safety in adults to do that before you can even move down into the pediatric age range. And even with that, often pediatric studies to move down into a 2 or 3 year old range is still usually something that's really difficult to do even with the first pediatric trial. We are involved in a lot of clinical trials in type 1 diabetes, and that's the other reason why we want to move up here at UCSD, because some of those studies are broader age range, so anywhere from age 6 up to age 35. I think a lot of people think that type 1 diabetes starts in childhood, and that's true, but most probably even maybe about 50% of patients actually develop type 1 as adults. And so this is not just limited to starting type 1 in the pediatric age range. And so we can improve our recruitment and our standing in the community by being able to draw both children and adults into these clinical trials. And the last reason, which was alluded to earlier, to bring everything together sort of under one roof, is really to provide the proverbial water cooler. Because science gets advanced and clinical medicine gets advanced by a two-way dialogue. It doesn't work by someone being in the lab and someone being in the clinic. And it's really that off-the-cuff conversation you have, some observation you made that triggers a thought in somebody else's mind and goes, well, yeah, and this is how then ideas get advanced. And you really have to have that ability. And unfortunately, right now, we're a little fragmented geographically. And so to be able to bring everything up to the UCSD campus, I think will be a tremendous benefit for the patients we serve, the community, for continuing research, obviously working with our colleagues that are in the bioscience. I mean, San Diego is so fortunate to have all this enriched bioscience environment that a lot of places don't have to that. So we want to take the maximum and maximize, as I say, sort of the diamonds that are in the backyard. We need to reap those and harvest those, and that's another power that we have at UCSD. So thank you for your attention, and I'd be glad to answer any questions.
Alberto.
All right, Elsie.
Paul, I really hate to ask you, but where is the beef of this presentation? I tell you why I said that. There are many people with diabetes in this room waiting to hear some results. I know that it's too early, but 4 patients are probably enough to get an idea of whether those cells are producing a little tiny bit of insulin. In a mouse, it takes 8 to 10 weeks before you get full insulin production to reverse diabetes. In a human, I assume that minimal amounts should be enough to at least encourage the people that are here saying, yes, it might work. Or now, a positive result in 1 patient will be sensational. A failure, you can make no comments. So my question is, if you have 6 patients, after 6 months, you should be able to say, it works, it's hopeful, but I hope that we won't have another meeting in a couple of months and say, well, we did 6 patients now and not much has happened.
Sure. Yeah, in this first group of patients, which, as I said, are quote-unquote subtherapeutic, mainly to look at safety. As Jeremy said, we are putting in these sentinels, and we are pulling those sentinels periodically and doing what's called histology, essentially going in and looking at the cells. And we can look at those sentinels to see, are they vascularizing, are the cells surviving, and are they differentiating similar to what we saw in the animals. So we will get that data. I don't have enough data or any data to tell you today. In terms of actual insulin production, we have some fairly sensitive assays, so we will certainly look. But if we didn't see it in this first trial, that wouldn't necessarily mean there's a problem, or in this first group of patients, because it is designed to be subtherapeutic, and there's a limited detection on the assay that we can use. But yes, we will look at that. And once we get enough patients in this first cohort completed, we plan to take all of that data to an independent data safety monitoring board. This is a group of physicians, experts in the field, that will review the data, certainly with an eye to safety, but they'll also be looking at what's called a futility analysis. So they'll be looking at the results that we have to date to decide whether or not it makes sense to continue exposing patients at higher doses after that. So that data safety monitoring board, once they've had ability to review the data and make their determination, that'll be the first real piece of information that will be out there to suggest that everything is going as we expect it to.
One more thing. Several years ago, Swedish people did a very neat experiment with human fetal cells. Sorry. Did you hear the beginning of my... Many years ago, Swedish investigators did a very neat experiment. They took human fetal pancreas, which is the equivalent of your progenitors from stem cells, and implanted under the arms in humans. And with 20,000 islets, or 20,000 islet-like structures, they were able to pick up C-peptide or insulin. But the neat experiment was that they put the cells in one side, in one forearm, and not in the other one. So minimal amounts could be detected in a differential way from one arm to the other one. I would suggest that you people think of putting one of these little capsules into one arm and then seeing if that small dose replicates the experience with human fetal pancreatic cells.
Yeah, I'm not familiar with that work, so I can't comment on, I don't know if others are familiar with it, but we've looked at, I can only assure you that I think we've taken a path that we hope, based on consulting with a lot of experts out there, is the one that we think gives us the best opportunity to not only evaluate the safety, but also during this time, keep in mind, as I said before, this is the first time this has ever been done. Anything like this has ever been done. And so the surgical techniques, the treatment of the patients, the aftercare, all are going to have impact on how well these cells graft in, okay? In other words, how well they survive the very traumatic environment that they're first put into, and then how well they then go on to continue to mature and differentiate. And so those are all things these sentinels allow us to spend time understanding. We meet every, as he can tell, we meet after every patient with the surgeons, with the team, and we discuss what went on, what we saw, how the patient responded. And as we get data, we'll start having more data to have further discussions on that to improve the techniques to make sure that we give this the best chance of being effective. But all I can ask is for patience at this point.
One last question?
And so in typical drug discovery and drug development, you usually start with normal, healthy volunteers, but you're talking about patients right away. Can you explain a little bit how you got there?
Yeah, well, it's typical in treatments like cancer treatment, in this case type 1 diabetes, where you have a fairly invasive therapy, to go directly into patients. And really, we wouldn't be learning the same, even the safety studies and such would not be that informative in a normal volunteer in this case. So there was no issue with the FDA when we proposed that go directly into patients. We're obviously starting slow and taking baby steps as we begin, but the idea was to go directly into patients in this trial. So we call this a phase 1-2 trial, okay, where you're not only looking for safety, but also have efficacy endpoints.
Okay, one more.
I want to tell y'all that your research has reached Texas. We came up from Texas to hear this, and we want to thank you very much for what y'all have done. My question is, do we have enough islet cells to produce for mass production?
So do we have enough islet cells? So there's a couple ways of answering that question. One is, can we produce enough cells in the manufacturing process? And the answer to that is yes. And that goes back to, as Larry said, stem cells in general. In our case, we start with embryonic stem cell, are what's called pluripotent cells. They are regenerative. They replicate. And they can go on to produce pancreatic cells and islets over time. And we have these banked. We have masters and working cell banks of this cell line that we are starting material, and we can take a vial out of a working cell bank with millions and scale that to billions of cells within a couple weeks. So it gives us almost an unlimited supply of that, essentially an unlimited supply of that starting material.
Can I ask the last question?
We have a few other people from the audience. Maybe the experts will also be here. We have another 20 minutes of, we can have a glass of wine, so you can also ask the questions directly. But there was a question here from...
How do we become patients?
What was that?
How do we become patients?
I'll turn that over.
That's a good question. So we're really just taking 18 to 55-year-olds, healthy men at this point, for safety reasons. And we really are still looking for people. There's a list of people that we've been kind of accumulating. I think that in this very early stages, it takes, realistic expectations, keeping in mind that, again, this is kind of for safety purposes. We've had people contact us from literally all over the world saying that they'll, fly in from Italy to get this implant or whatever. And keeping in mind that, the implant is step one. And then it's following you, there's multiple visits, a lot of time requirements, things like that. So, if people are interested in our local, then we definitely can take names and things like that. So we'll be here afterwards.
And I should point out that the selection of patients is totally with UCSD.
Totally with UCSD, yeah. So there are some, basic requirements, but also, it takes keeping a special eye for people that, are on top of their diabetes and willing to kind of undergo all these different procedures, and keeping in mind, again, that this is looking at safety.
And I do have a microphone in my hand, so I'm going to ask my question. I actually have these devices in my back right now, so if you guys want to ask me about how the clinical trial is and what it's like to be in it, feel free. My question, though, I do have a real question, wasn't just announcing that, is since they are pancreatic precursor cells, they can become alpha, beta, any of those Greek letters. What is the fractions that we've seen in preclinical models, and sort of, are there any worries about production of other polypeptides?
You want to take that one?
Yes, you're correct. And by the way, thank you for participating. We, by the way, at the company have no knowledge of who the patients are. It's good clinical practice that we don't have that knowledge. So, so this is the first time I've been here for that, but thank you for participating. It is as we start with the pancreatic progenitor, it's a mixture of immature endocrine cells, which are the cells that go on, that are produce insulin and such. As they mature, it looks very similar to an islet. There's actually, I should have probably mentioned this at the beginning, there's a good clinical proof of principle for this idea of doing a cell replacement therapy for type 1 diabetes. There's a procedure called the Edmonton protocol that uses cadaver islets. And they take those cadaver islets and they implant them in the liver portal vein. And this is done in very, very ill patients, at very high-risk patients. It requires a the life of the implant immune suppression. So it's a very difficult procedure and not one that is used very often, but there's about 1,000 patients that have received it, and it is effective at controlling the disease. And so we can look at that and we can look at the implants and look at the beta cell mass, the alpha cells, which produces glucagon, and delta cells, which produce somatostatin, and we can get an understanding of what we need to have in our graft. And it looks very similar, okay, to what you see in a typical islet. You know, if anything, a bit more like a kind of a young islet, immature islet, a little bit more somatostatin. But in answer to the key part of your question is, you know, is there any issue with, you know, how much you have there? We have never seen hypoglycemia in animal models, even when we've given 30 times the cells that we plan to use in a patient. And this, we believe, is because these are exquisitely regulated like a normal beta cell. They detect glucose level in the blood. In fact, when we put these in a mouse glucose is about 160 milligram per deciliter. So those of you who are familiar with type 1 diabetes know that that's kind of hyperglycemic, okay, from a human standpoint. Human, you're shooting for about 70 to 100 milligrams per deciliter. When we put these cells into a mouse, even when they have an intact mouse pancreas, they regulate the mouse down to the human level. Okay, the mouse will now become an animal with a glucose level of about 100 milligrams per deciliter, and it stays that way for the life of the mouse, or until we pull the cells back out, and then they go back up to 160 milligrams. And the reason I tell you that is it just shows how exquisitely regulated these cells are. They're able to detect the level of glucose in the blood and then release the insulin that's appropriate for the set point that they are seeking.
This first cohort, we've increased it up to about 10 patients, give or take, with the idea that we'll increase it to the second cohort, which will be multiple sites, up to around 40 patients. But again, this first group is only at UCSD, and now we're going up to, again, about 10.
This is more on a business type of question for Paul. So what you guys are doing are more biological cure. The race for the cure has been there for years. Like you said, 20 years ago, everybody's racing. Two-part question. Two-part question. Artificial pancreas, more medical device race for the cure, or not even a cure, but management, versus how much lobbying have you been getting against what you're doing from the Novo Nordisks, from the companies that are making a lot of money?
The easy answer is none. In fact, our largest single investor in our company is J&J Development Corporation, which is the investment arm of Johnson & Johnson. And we have been certainly approached by other companies with interest in what we're doing. So, you know, companies recognize they may make a lot of money out of insulin and that type of thing, but, you know, they know that the technology moves forward and they want to be part of the next stage and to continue to develop and treat this disease. So, you know, everybody recognizes if you come up from a business point of view, if you come up with a cure for a major disease, that's probably not a bad business proposition. Okay, so, you know, we haven't seen any pushback on that.
What about the artificial pancreas?
So artificial pancreas, yeah, it's interesting. I was giving a talk up in Silicon Valley a couple weeks ago, and they had 3 speakers. One was talking about an application that brings data together for type 1 diabetics that can really help to manage the disease. So that's kind of right now, a treatment, you know, something you can do right now to it. The second speaker was artificial pancreas, and the data was really exciting to see. For those of you who don't know what artificial pancreas is, it's essentially marrying a insulin pump with a glucose sensor with a smart computer controlling the amount of insulin, and there's also potential for glucagon infusions and such to control that and better manage the disease. And, you know, the speaker for this from Harvard, he was talking about it, and he showed his data, and then I was up next. He said, now you've heard what we're doing and what we think this could be in a few years. Now let me introduce the person who's going to come up here and tell you what we can do in the long term to really control this disease. And that's how we look at it. The insulin, we applaud the work going on with the artificial pancreas and think it's very exciting. But I think we all would like to have something that we treat and quit worrying about it.
So I think with this, we want to close here tonight, and we had a wonderful discussion. And I think one thing that this trial really exemplifies is that really it's sort of a long road before you actually get to something that is really this game changer for a disease treatment, and that you really cannot shortcut. You know, the safety of the patient is important. We need to know we're doing the right thing, and things need to be done properly. And the only way then to really do that is to really look and do the best science, and then to translate that into the best medicine. And there's nothing that this trial really exemplifies that. It's literally 30 to 40 years of work that went into this trial. And that is something to really keep in mind, that people have started to lay the foundation for what we're now seeing to go into patients already in like 1980 and 1985. So all that knowledge went into that. And I think that's also important to keep in mind, that we need to continue to invest into the basic science and to continue to keep up the hard and the good work. And San Diego is an awesome place for that. And just to maybe give you one number that really exemplifies that, when you take federal research dollars, and some of you might have heard how hard are those to come by, that really only the best are actually getting the money from the federal government to do research. And when we look at federal research dollars per faculty member, UC San Diego is #1 in the nation. So that just gives you an idea how good of an environment we actually have here. And then we have fabulous industry partners. We have a very vibrant community for biotech. So there's really no better place than here to really make this happen, and then maybe see this therapy come to fruition, and maybe other things that can actually help improve the lives of people with diabetes. And we hope to really accomplish that in this comprehensive diabetes center that we are building here at UCSD. And with that, I want to close, and I want to really encourage you to introduce each other to each other. You can also find the experts. We still have wine and cheese out, so stay for a little bit. We have some warming lamps, so stay warm. And thank you so much for coming here. It was a real pleasure.